The Earth is a complex ecosystem. Just as plants and animals affect each other and the environment, microbes, the beings we can only see with a microscope, affect humans and the ecosystem in important ways. Microbes, as their name suggests, are microscopic and invisible to the naked eye. How then do we know how many and what kind of microbes surround us?
You can take an air sample and culture it, and maybe after 48 hours, they will tell you, oh, there are viruses in that room. By that time, you have had all your coughs and colds and everything done.
Professor Soumyo Mukherji and his team are working on low-cost, highly effective biosensors that can detect and identify many of our microscopic co-inhabitants.
The sensors help us in determining rapidly a parameter that we want to measure. Let's say bacteria in water. Typically, the people will take a water sample, send it to a lab, culture it 48 to 72 hours. Our sensor does it in half an hour, and that goes for everything. Or antibiotics in water. You want to do it using a LCMS. It will take you 3,000 rupees per sample. You want to do it using the sensor that we have developed, 500 rupees per sample. Wide-scale deployment depends on cost, ease of use of an instrument, and such parameters.
Professor Mukherji's lab has improved their device over the past 12 years. It can be used to detect a variety of microbial life.
We have developed sensors for bacteria, viruses, sensors for heavy metals in water, for antibiotics in water, in milk, food, meat, you name it, we can develop a sensor for that.
Many of the microbial life that surround us are pathogenic, meaning they pose a threat to our health and our lives. So it becomes pertinent to disinfect our surroundings, and Professor Ambarish Kunwar's lab has been developing devices that use ultraviolet light to achieve this.
With the support from the WRCB at IIT Bombay, we were able to develop two different kind of ultraviolet disinfection robots. One of them was a cylindrical unit, so basically it's an arrangement of 6 UV lights mounted in a circular fashion on a robotic platform which can be controlled wirelessly, and this was something which we were able to assemble in about 40,000 rupees at a much cheaper price than what it was available in the overseas market. And then we also designed another robotic distribution unit which had an arm which could fold, unfold, could move up and down, and could access all the areas which were not accessible by the traditional robots, ultraviolet disinfection robots. We also had a Biorak project, and using this project we are developing a far UVC based tunnel for the disinfection of the people who are passing through the tunnel.
As we dive deep inside the microbial world, we are in for some surprises. Giant viruses, relatively recently discovered organisms we do not know much about, are one of the subjects Professor Kiran Kondabagil researches.
So when some viruses are in excess of about 200 nanometers in size and carry a genome of about 300 kilobases, these are all what we call as giant viruses. Unlike most other viruses, they don't depend on the host cell machinery for copying more of themselves.
Some giant viruses are bigger than bacteria. Scientists have found giant viruses in various environments such as oceans, lakes, forest soils, and the human gut. They speculate that these viruses contribute towards maintaining the ecosystem.
We are trying to look at not only how these viruses are actually surviving and how big they are, we are also looking at what we can learn from the sheer diversity and numbers and how they can impact the environment around them.
Professor Kondabagil has isolated several giant viruses from many places, including sewers. Among them are mimiviruses that infect amoeba. He and his team are studying the key proteins of these viruses that allow replication in the amoeba. While pathogens are everywhere, not all microbial life is a threat to humanity. Some are harmless, while there are a few which are even beneficial to humans.
Our basic interest is to look and isolate new microbes which has a better ability to degrade the aromatic pollutants like naphthalene, methyl naphthalenes, various aromatic pesticides like 2,4-D carbaryl.
Professor Prashant Phale identified a new species of pollutant-eating bacteria and named it Pseudomonas bharatica CSV 86. He discovered them in soil and isolated and purified them. These bacteria preferentially consume naphthalene over glucose.
We thought of engineering this strain where we can not only degrade naphthalene, but can we add metabolic pathways which can give the robustness and metabolic diversity to degrade these aromatic pollutants better way.
Professor Phale and his team are genetically engineering bacteria to clean up more of the pollutants than what they are naturally capable of. For this, they used a genome sequence from a carbaryl degrading strain and created a new strain that could degrade naphthalene and its derivatives as well as carbaryl. The team is currently exploring organisms that can break down plastics, remediate lands, and also promote plant growth. At BSBE, IIT Bombay, researchers are not only exploring and discovering these microbes, but also understanding them better, detecting them, protecting us from them, and even putting some of them to work for our benefit.